Yes, all right. So, in today's session, I was saying that we will be focusing on the morphology and structure of the microorganism. In the previous class, you've studied about the introduction to the microbiology, in which you have studied about the different types of microorganisms that are present in the surroundings and environment, such as bacteria, viruses, parasites, protozoa, and fungus. You have also studied about the microscopes as well. But in today's session, it is mainly focused on the morphology and structure of the bacteria and the fungus. So in today's session, we'll be talking about the structure of the bacteria and fungus. All right. Yes. So starting with bacteria. So what are bacteria? So bacteria are the prokaryotes. They come under the prokaryote cell. There are two types of cells. First one is the eukaryotic and second one is the prokaryotic. So what does prokaryote means? Pro means before and kary carry means nucleus. So before nucleus. So which the organisms which does not have the well developed nucleus or the nuclear membrane are comes under the class prokaryotes. All right. So the prokaryotes are the organisms that do not have the well defined nuclear membrane. So we can say that the bacteria are the prokaryotes because they do not have any kind of nucleus or organelles bound in the membrane, the nuclear membrane. So they are most ubiquitous in nature. They are generally single cells. Most of the bacteria are single cells, but some may be multicellular also. All right. So they constitute a large domain of the prokaryotic microorganisms. All right. So they are typically a few micrometers in the length only, and they are the first life forms that has appeared on the earth. All right. So DNA of the bacteria is a single circular chromosomes. No histones are there which are associated with the DNA in the bacteria. All right. Now talking about the cell wall of the bacteria. So if we talk about the cell wall of the bacteria, it is made up of the peptidoglycan layer. All right. We'll be studying about in detail in today's session. The smaller ribosome units are there and they divide by the binary fission and not by the mitosis. So we can say that bacteria are the single cellular microorganisms which are prokaryotic and they have a DNA which is a single circular chromosome. They are a microscopic organism which cannot be seen with the naked eye. This is clear. The concept of bacteria. Now talking about the size of bacteria. What size of the bacteria? All right. So average bacteria size is 0.5 to 2 micrometer in diameter. If we talk about RBCs, is 7.5 micrometer in diameter. So it is very small than even the RBCs, which is the smallest cell of the blood. The surface area of bacteria is 12 micrometers square. All right, the volume is 4 micrometer square. And the surface area, if we talk about the ratio of surface area to the volume, it is 3:1. Typical eukaryotic cell surface area to volume is 0.3:1. You have to learn these things. All right. So you just have to learn this thing. There is no shortcut to this. You have to learn. All right. So this is about the the prokaryotic bacteria, but eukaryotics needs structure and organelles, but there is nothing with there's nothing in relation with the bacteria. So if we talk about the size of the bacteria, average bacterial size is 0.5 to 2 micrometer in diameter. This is the most important thing. Now talking about the shapes of bacteria, that what shapes of bacteria are there. You can see the different shapes of bacteria there. All right. So first of all, bacteria are classified into different classes on the basis of their shapes. So there are different classes such as cocci. Cocci are the ones which have round shape. All right. So bacteria which are of round shape are called as the cocci or coccus. All right. So in cocci, there is chain-shaped bacteria, which is called as the example of which is a streptococcus. The streptococcus bacteria are the round-shaped bacteria that have the arrangement in the form of chains. So they are arranged in chains, beads-like structure in the neck piece, such as. So the beads-like structure in the chain form is the streptococcus. Next one is the cluster formation. So the round shaped bacteria which are there in the cluster formation or grape like formation they have are the example is staphylococcus. Then next is the bacilli. First class of the bacteria on the basis of their shape is cocci. The second shape of the bacteria, second class of the bacteria is the bacilli. All right. So what are bacilli? Bacilli are the rod-shaped bacteria. So bacteria which have rod-shaped are termed as bacilli. All right. So bacilli appear in chains. The bacilli that appear in chain form are the streptobacillus. Then there is one more thing, that is cocobacilli. What is cocobacilli? The bacteria is bacilli shaped, but it is not elongated in the length, but it is narrow and it has rounded diameter. So the bacteria that has a rounded structure but in the rod shape form comes under the cocobacilli form. All right. So this you can see, this is the cocobacilli. It is round but elongated also. But the length is not much elongated as in the case of bacilli. All right. So this is the cocobacillus. Then the next shape of the bacteria is the vibrio. All right. The example is vibrio, but the bacteria is curved shape. If the bacteria is curved or comma shaped, this is the classification of bacteria of comma shape, which is curved bacteria. The example is vibrio. All right. Next is spirillum. So the bacteria which have spiral shape, or which you can see here in this picture also, that the bacteria which have coils in it, we can term it as spirillum. Next is spirochetes. So spirochetes are the bacteria which have a lot of coils. You can see there is a lot of coiling in the bacteria. So this is spirochetes. Next classification is square. So the bacteria which is square shaped, it is termed as square. There is one more star. So these are the shapes of bacteria. Bacteria can appear in different shapes. All right. So the different shapes of bacteria are this. The cocci ones, which are round shaped bacteria, the bacilli ones, which are rod shaped bacteria, then the cocobacilli, which are round shaped or but a little elongated at the ends. Then there is curved shaped or the comma shaped, which is Vibrio cholera. All right, students, you don't need to write things in the chat box. You just have to answer the questions that I have asked to you in the chat box or whatever related to the content only. Now talking about the structure of the bacteria, that what is bacterial structure. So if you will see the anatomical structure or if you will see the internal of the cell, you will be able to spot these structures in the bacterial structure. So what are these? Number one, there is you can see here the thing that is there. This is the accessory thing. This is not found in each and every bacteria, but in some of the bacteria. This is flagellum, right? There is there is a capsule also which is present in some of the microorganism, but not all of the bacteria possess it. All right. Then there is the cytoplasm. Then the cell membrane is there, which is cell wall is there. Then the cytoplasmic organelles are suspended into the cytoplasm of the bacteria. There are ribosomes, the inclusion, the Golgi bodies. All right. Then there is nucleus also in some of the bacteria. So the bacterial structure consists of flagella or pili, the capsule, the plasma membrane, cytoplasm, cell wall, which can be made up of the lipopolysaccharides, teichoic acids may be there, inclusions are there, and in some of the bacteria, spores are also present. So this is the bacterial structure. You are able to understand this bacterial structure, students? You're able to understand this bacterial structure, students? So the bacterial cell is surrounded by a cell membrane, which is made up of primarily of the phospholipids. This membrane encloses the contents of the cell and acts as a barrier to hold nutrients, proteins, and the other essential components of the cytoplasm within the cell. Unlike talking about the eukaryotic cells, bacteria usually lack their large membrane bound structures in the cytoplasm, such as nucleus, mitochondria, chloroplast, and the other organelles present in the eukaryotic cells. All right. But some of the bacteria have protein bound organelles in the cytoplasm, which what will it do? Which will compartmentalize the aspects of the bacterial metabolism, such as the carboxysome. All right. So additionally, if we say bacteria have a multi-component cytoskeleton to control the localization of the proteins and nucleic acids within the cells and to manage the process of cell division. All right. So bacteria do not have a membrane bound nucleus, and their genetic material is typically a single circular bacterial chromosome of DNA, which is located in the cytoplasm in an irregularly shaped body, which is called the nucleoid. All right. So the nucleoid consists of the chromosomes, all right, with its associated proteins and the RNA. All right. So, like all the other organism, bacteria contain ribosome for the production of proteins, but the structure of bacterial ribosome is different from that of the eukaryotes and the archaea. All right. So, around the outside of the cell membrane, if you will see in this picture, is the cell wall. So, bacterial cell walls are made up of the peptidoglycan layer, which is also called as the murein. It is made from the polysaccharides chains cross-linked by peptides containing the D amino acids. All right. So these the bacterial cell walls are little different from the cell walls of plants and fungi, which are made up of chitin and the cellulose respectively. All right. So the cell wall of bacteria, if we say, is also distinct from the archaea also. So, if we broadly speak about the two different types of cell wall in the bacteria, they are classified into gram-positive bacteria and the gram-negative bacteria based on the presence of the components inside the bacterial cell. All right. So let's talk about the different structures in detail. Uh, First one is the flagella. So flagella, what is there? Flagella is present in some of the bacteria. It is not present in all of the bacteria. Flagella is there for providing the motility or movement to the bacteria. So flagella, because of the flagella, the bacteria is able to move. In some of the bacteria, swarming also occurs because of the bacteria. The bacteria are moving like this, and you can see with the help of some of the reaction that swarming of the bacteria is there. Then arrangement basis for classification of the bacteria is there. So on the basis of flagella, we can also classify the bacteria into different types. There is monotrichous flagella, lopotrichous flagella, amphitrichous flagella, and the peritrichous flagella. So what is monotrichous flagella? Mono means singular. So if the bacterial cell have only a singular flagella, a single flagella is there, then we can say that the bacteria has a monotrichous flagella. Then the lopotrichous. What are the lopotrichous bacteria? Lopotrichous bacteria are the ones that have a tuft of bacteria, a group of bacteria at the end of the bacterial cell. All right. Then the amphitrichous bacteria. Amphitrichous, the bacterial cell will have the flagella at both of the ends. Then the peritrichous bacteria will have the flagella all around the bacteria. All right. So here you can see the mono or lopotrichous. So mono is the ones which have a singular flagella at one end. So they have the circumlocation, circumentary location, rotation. Then the This is the peritracheous bacteria, which have the flagella at all over the end. Then this is the lopotracheous bacteria, which have a group of flagella or tuft of flagella at one end. All right. Now talking about the pili. So what is pili? So pili are the short protein appendages which are present in the bacteria. These are smaller than the flagella. What is their function? The function of the pili, the main role of the pili is providing the attachment of the bacteria to the host cell or any other thing. All right. So the adherence is provided by the pili. If we talk about the example, E. coli has numerous types of pili in it. The antibodies will block the adherence, but the F. pilus is also used in the conjugation process and the exchange of genetic information in the bacteria. The pili also provides the flotation, which will increase the buoyancy of the bacterial cell. All right. Now talking about the capsule or slime layer. This is again an additional component of the bacteria. It is not present in all of the cells. But what capsule do? Capsule provides the pathogenicity to the bacteria, increased pathogenicity to the bacteria. So what will it do? It will provide the adherence to the bacteria to adhere it properly, and as well as the antibiotics or the antibacterial things will not be able to enter into the cell wall easily because of the presence of capsule or slime layer on it. It is the capsule or slime layer is made up of the glycocalyx or the polysaccharide on its external surface. What does capsule or slime layer does? It prevents the phagocytosis. The cells of the body's immune system will not be able to phagocytose the bacterial cells easily due to the presence of capsule in it. Thus, it is increasing or enhancing the pathogenicity or virulence factors of the bacteria. All right. Next is the cytoplasm. So cytoplasm is 80% of the water, and 20% of it is because of the salts or protein in it. So the cytoplasm prevents the osmotic shock. All right. So the DNA is circular in it, and it is haploid in nature. Mutations allow adaptation to environment quicker. Plasmids are also present, which is extracircular DNA, which provides the antibiotic resistance to the bacteria. No organelles such as mitochondria or Golgi is present in the bacteria. So the cytoplasm of the bacteria is made up of 80% of water and 20% of salts or proteins. And some of the inclusion bodies are there in the cytoplasm of the bacteria. Now talking about the cell membrane. So cell membrane of the bacteria is a bilayer phospholipid. It is made up of the phospholipids, two layers of the phospholipids. The water can easily penetrate into the cell membrane. It is flexible in nature. It is not very much strong. It can rupture easily. The osmotic pressure is created by the cytoplasm in the cell membrane. You can see the structure here. So it consists of two regions. We can say first one is the hydrophilic region, in which the water can be attached, and second is the hydrophobic region, in which the water cannot attach. The fatty acids are present and glycerol are present in the hydrophobic region, which makes it insoluble to the water. All right, it is attached with the phosphate group. Here you can see the structure. The lipid bilayer membrane is there. It is made of the phospholipid molecules. Hydrophilic ends and hydrophobic ends are there. Then the glycoprotein are also present in the bacterial cell. The glycolipids are there, and extracellular space is there. The internal membrane also attaches cholesterol, protein, and cytoplasm. Now talking about the cell wall. The cell wall of the bacteria is the most important structure in the bacteria because the mechanism of the pathogenicity depends on the cell wall of the bacteria. So the cell wall and the identification also, and thus the treatment also. So the cell wall is made up of a peptidoglycan polymer. It is made up of amino acids and sugar. It is also called as the murine sometime. It is unique to the bacteria. It consists of N-acetyl glucosamine and N-acetyl muramic acids. The D form of amino acids used, not L form. This is hard to break down the D form. So on the basis of cell wall also, the bacteria are classified into two very important classes, which is gram-positive bacteria and the gram-negative bacteria based on the components of the cell wall. We'll be talking about it in later. All right. So you can see here the cell wall of the bacteria. It is made up of N-acetyl glucosamine and N-acetyl muramic acid. The lysozyme sensitive bonds are also there. It is attached with the peptide bonds. You can see there the CONH bonds are there. All right. So this is the biochemical structure of the cell wall of the bacteria. All right. So here it is given the cell wall. All right. So this is the gram-negative membrane, and the periplasm is there. Peptidoglycan layer is there. Cytoplasmic membrane. So what is the basic difference between the gram-positive and gram-negative bacteria? Let me tell you. In the gram-positive bacteria, the cell wall has the thick peptidoglycan layer, which is closely packed. But as you will see in the gram negative membrane, it has a peptidoglycan layer, but it is thin. And the cells in the peptidoglycan layer, you can see here, are loosely packed. These are not closely packed. You can see these smaller structures. These are cells which are loosely packed. All right. The gram-positive bacteria also consists of teichoic acids. All right. So the teichoic acids present in the cell wall of the gram-positive bacteria increases or enhances its peptidoglycan layer and its retain property to the primary stain. Whereas the gram-negative bacteria is absent of the teichoic acid. All right. So here you will see the peptidoglycan layer is thick and the cells here in this peptidoglycan layer are loosely packed. It also consists of the lipoprotein, the phospholipids, the lipopolysaccharide layer is there, which consists of porine, protein, O polysaccharide, and the core polysaccharide. Outside, from outside it looks like this, and from inside it looks like this. Now talking about the teichoic acid. So the teichoic acid is present only in gram-positive bacteria. It consists of glycerol, phosphate, and the ribitol. It provides the attachment for the phages. All right, here you can see that the peptidoglycan layer in the gram-positive bacteria is thick and the cells are closely packed in it. It consists of teichoic acids and the lipoteichoic acids, which provides the attachment for the phages. So what it do? It provides the protection from the phagocytosis. All right. Now talking about the lipopolysaccharide. Lipopolysaccharide layers are present in the cell wall. So it is endotoxin or pyrogenic in nature, which is fever causing, you can say. It has a toxin nomenclature. The endo means part of bacteria, inner part of bacteria, and exo means the outside or into the environment. So the lipopolysaccharide consists of a lipid polysaccharide and the G bacteria only. It removes the alcohol or acetone from the bacterial cell. All right. So what is the function of the lipopolysaccharide? It kills the death of the bacteria is due to the LPS. It is pyrogenic in nature. It can cause fever. So we can say it is what it does. It provides or enhances the pathogenicity or virulence of the bacteria. It is resistant to heat and it is very hard to remove. All right. This is about the lipopolysaccharide. Now talking about the endospores of the bacteria. So endospores are the resistant structures. These are extra structures which are present on the surface of the bacteria. They are resistant to the heat, irradiation, and the cold, and they also resist boiling for up to 1 hour, and they can be still viable. All right. So the location is important in the classification, the spores location. So endospore can be centrally located, they can be subterminally located, and can be terminally located also. All right. So they are used for the quality control of heat sterilization equipment also, to see if the equipment is able to destroy all these spores or not. So the spores are destroyed only by the process of sterilization, and they are not destroyed by the process of disinfection. All right. So now talking about the fungi. What are fungi? So fungi are the ubiquitous and diverse group of organisms. The main purpose of fungi is to degrade organic matter. All right. So this is the bacterial part is clear to you, students. Please tell me if the bacterial part is clear to you. All right. So fungus is any member of the group of the eukaryotic organism, which includes microorganisms such as yeast and the molds, as well as the more familiar mushrooms. So these organisms are classified as a kingdom separately from the eukaryotic organism. There is a separate classification of the kingdom for the fungus, which is fungi, which is fungus. All right. So, by one traditional classification, including Plantae, Animalia, Protozoa, and the Chromista. All right, so a characteristics that places fungi in a different kingdom from plants, bacteria, and some protists, chitin in their cell wall. The fungus consists of the chitin in their cell wall. So fungus are heterotrophs. They acquire their food by absorbing dissolved molecules, typically by secreting digestive enzymes into their environment. So fungi do not process their food by the process of photosynthesis. Instead, they degrade the organic matter and create their own food. Thus, we can say that fungus are heterotrophic in nature. All right. So growth is their means of mobility, except for spores, which may travel through their air or water. So fungi are the principal decomposers in the ecological system. These and other differences place fungi in a single group of related organism named the Eumycota, which is true fungus. All right. So they are abundant worldwide in nature. Most fungus are also inconspicuous because of the small size of their structure and their cryptic lifestyles in soil or the dead matter. Here you can see they occur as a heterotrophic existence as four forms: the saprobes, the symbionts, the commensals, and the parasites. We'll be talking about all of these classification in the later section. So as a heterotrophic, fungus can occur in all of these four classes. Now talking about the characteristics or the properties of the fungi. So habitat, if we talk about what is the habitat of the fungus, so they are ubiquitous. They appear in terrestrial land, the terrestrial areas also, such as lands and the fresh water also. If we talk about the distribution, their distribution is cosmopolitan. They are everywhere. All right. If we talk about the ecology, so important ecological role they play, such as the saprotrophs or the mutualistic symbionts or the parasites, providing the mutual benefits to the symbionts. It also decomposes the organic matter, so by existing as the saprotrophs. If we talk about nutrition, I told you that the fungus are heterotrophic in nature because they compose their own food by the degradation of the organic matter. Vegetative state, non-motile myceliums are there. The cell wall is composed of glucans and the chitins. So if we talk about the nuclear status, it consists of uni or multinucleate. There can be a singular nucleus and there can be multinucleus also. The thallus being homo or heterokaryotic. They may be haploid, dikaryotic, or diploid also. If we talk about the mode of reproduction in the fungus, there is both of the modes of the reproduction are there in the fungus, which is sexual reproduction. For example, it can produce the reproduce by the nuclear fusion and meiosis, and the asexual reproduction also. This is the internal structure of the fungal cell. You can say, see the anatomy of a fungal cell. It consists of cell wall. Inside the cell wall, there is this vacuole and endoplasmic reticulum are there. Endosomes are there. Cell membrane is there, which then mitochondria is there. Golgi bodies are there, and the secretory vesicles are also there. So this is the anatomy of a fungal cell. The cell wall of the fungus consists of glucans and the chitin. Talking about the morphology or shape and size structure study. So fungi can be unicellular, like a singular cell may be present, and multicellular, multiple cells may be also there. They appear either as yeast or molds. Yeast or molds are the most important forms of the fungus. The yeast are usually unicellular in nature, whereas molds are multicellular organism which consists of thread-like tubing structure which are hyphae. So here you can see the yeast cell. The yeast cell is large. It is 5 to 8 micron in nature, micron in size. It is single cellular or celled organism which rarely form filaments. Most yeasts produced by the asexual process of budding. You can see here the picture of the yeast cell. It consists of the secretory vesicles, fat globules, Golgi, vacuole, cell membrane, cell wall, mitochondria, endoplasmic reticulum, and the nucleus. Now talking about the classification of fungus, that how does the fungus are classified. So there is kingdom, then the kingdom is sub-classified into subkingdom, then there is phyla, then subphyla, class, order, family, genus, and the species. So the fungus are classified into four major classes, which are ascomycetes, basidomycetes, phycomycetes, and the deuteromycetes. So the class ascomycetes reproduce by the process of sexual reproduction in a sac form. The sac form, the covering form, is called an ascus. Thus, they are termed as ascomycetes. Example of the ascomycetes is the aspergillus, which is very important fungus, pathogenic fungus. Then the second one is the basidiomycetes. So the classification of these fungus is based on their modes of reproduction, that how they reproduce. So the ascomycetes reproduce in the form of sac. So it is the sac, the structure, the covering is called as ascus or sac, thus they are called as ascomycetes. Then the second one is the basidiomycetes. They reproduce in a sac called basidium with the production of basidiospores. Thus, they comes under the class basidiomycetes. The example of basidiomycetes is the cryptococcus. Then the phycomycetes. So phycomycetes can reproduce by gametes and asexual reproduction with by forming the zygospores or the oospores. The asexual reproduction with production of the sporangiosperms is there. Then the deuteromycetes are there, which are also called as the imperfect fungi or the fungi imperfecti. Because there is no recognizable form of sexual reproduction in the deuteromycetes. They include the most pathogenic fungi, which is the Candida. Now talking about the characteristics of the fungi or the properties of the fungi. So the ascomycetes have septate hyphae. They produce asexually. The characteristic spores are blastospore, conidium. Then the origin of spore, they produce by budding. Then the example is aspergillus or penicillium. Next is they produce pathogenicity or conditions like aspergillosis or mediromycosis. Then the basidiomycetes have septate hyphae. They produce sexually. They have basidiospores in it. The origin of a spore is basidium. Then the example is cryptococcus, and the condition caused by it is cryptococcosis. Next is deuteromycetes. Deuteromycetes has separate hyphae. They produce asexually. They have thallospore or conidium. The origin of thallospore is thallus or hyphae. Conidophore. The example is Candida. Most mycosis is caused by the Candida, fungi and pathogens in the medical imperfectly. Then the phycomycetes have aseptate hyphae. They produce both bisexually and asexually. By asexually, they produce sporangiospores. If they are reproducing sexually, they produce zygospore or the oospore. All right. So the origin is sporangiophore in case of sporangiospore, and in the case of zygospore or oospore, the origin of spore is fusion of the nuclei. Example of the phycomycetes is Mucor rhizopus, which produces the pathogenic condition mucormycosis, which is very much pathogenic in human beings. It can also provide the blindness of the eye or the black fungus disease. Now, what is the importance of fungi? So, fungi are most important group of organism on the planet because they produce the degradation of the organic material, which is very important for the life cycle to continue. So, they are important in many of the ways. First one is the recycling. Recycling is done by the fungus. Then the mycorrhizae and plant growth is provided by the fungus. It is important as food because it produces the mushrooms are mushrooms comes from the fungi only. Then as medicines they can also be used. They can use for the biocontrol processes. The diseases of the crop can also be prevented from these funguses. They can also stop the food spoilage. So this is the importance of fungi. So that's all about today's session. If you have any doubts or queries related to today's session, you may ask in the chat box. The chat box is now available to you all students. So if you have any queries or doubts regarding today's session, you may ask in the chat box. I am here to answer all your queries and doubts. Students, if you have any query or doubts regarding today's session, you may ask in the chat box now. Everything is clear to you? The fungus, the bacteria, the classification, the morphological structure. Yes, all right. It is clear to everyone. Yes, you understood. Okay. All right, students. So that's all for today's session. Now you may leave the session. You may end the session. If you still have any query, there is a feedback section provided on your LMS. You can ask your queries in the feedback section also. All right, students. Thank you for joining. Keep joining your online sessions on time. There is a webinar schedule on your LMS. The notifications of it is provided on your LMS. Please check your LMS time to time so that you get aware about all of the notification or the new notices that are there on your LMS. All right. Thank you for joining, students. That's all for today.